Welcome, guys. This is after long days. In this particular total video demonstration, we'll be talking about immunology. The immunology is a subject. In this particular part, you must learn the basic concepts of immune system and how the immunology work in a whole. Now, in these videos, from these videos, we'll be talking about each of the individual part of immunology, but after that I'll be making two of the videos which will cover whole immune system in a whole or in a big picture. Now, to understand big picture is the most important part about the immunology, because if we are learning the small part, smaller detail of the immune system, it could be become very much puzzling to you, and you need to connect all these things together. If you can't connect all these things, then you shouldn't be able to understand what is actually going on, because all these processes we are going to know is going on at the same time inside our body, right? So let us begin. Now, the first person to whom we need to give our privilege is this man, Edward Jenner. Now, he was the developer of the vaccine for smallpox. Remember, still nowadays smallpox is irradiated, but still at those time when the smallpox was a devastating disease, at those time he was the first person to develop the vaccination technique. We'll be learning more about vaccination in our future videos, but now let's go on. Okay. Now, if we begin here, sorry. Okay. Now, if we begin here, we can find that during the developmental stages, from the zygote, that means we are having two gametes, one male gamete, one female gamete. So we are having male gamete, and we are having also female gamete, and these two gametes are fused during fertilization to give rise to this zygote. Now, this zygote will start dividing the cell. This becomes a only single cell. So this is a cell, single cell. Now this cell start to divide, and this division will give rise to morula, then blastula, then gastrula, these different stages. Now a lot of cells in the blastocyst form, then another more cells in the gastrula form. Now in this gastrula form there are three kinds of cell linings are produced. One cell is called the ectoderm cells, or the outer layer cells. These are the external layer cells. Then the in mesoderm or middle layer, then the endoderm or the internal layer. The ectoderm layer will give rise to skin cells or epidermis, now neuron of brain, and also pigments. On the other hand, mesoderm will give rise to the middle line, like cardiac muscle, skeletal muscle, red blood cells, and smooth muscles in gut. And finally, this endoderm cell will give rise to thyroid cells, pancreatic cells, these different organs inside our body. Okay. Now in this particular phase of gastrula, those cells will become differentiated. Okay, at the beginning the cell was only one. Now that cell can give rise to many different types of cells. Now those cells will be differentiated into their desired cell. Now at the beginning all of the cells started their journey from a single zygote. So this cell is called the embryonic stem cell. Now this stem cell in the blastocyst condition can have the ability to be differentiated into many different types of cell. So embryonic stem cell can give rise to a red blood cell. It can also give rise to epidermal cell of our skin. It can also give rise to pancreatic cells. Okay, so all of these cells are having the potency at this stem cell actually having the potency of giving rise of many different types of cells. That's why these stem cells are called totipotent stem cells. Okay. Okay, now after some time this differentiation is going on, and then different types of cells are produced in different nature. Now how they establish this process? Because each and every cell contain their nucleus, and the nucleus is consisting of nucleolus and chromosomes. Now the chromosomes, all of them are having 23 pairs of chromosomes, remember. But all of them having these 23 pairs of chromosome, one set is from their father, one set is from the mother, which was from both the gametes. But what is happening actually, all of the chromosomes are having same number of genes, same types of genes, but it depends upon the expression of that gene, that whether the cell is going to be a red blood cell or it is going to be a brain cell. Okay, so that's the differentiation. Now here the different layers like ectoderm, mesoderm, and endoderm that gives rise to different layers. So remember, this is a very important part for the question of CSI and NET. Now you can find the ectoderm can give rise to epithelial cells, it can give rise to nervous system, sensory receptor system, and all this. Mesoderm can give rise to the reproductive system and excretory system, muscular layer, and all these things. Endoderm will give rise to different organs, or say urinary bladder, urethra, liver, pancreas, thymus, and all these things. Okay, now let us talk about the immune cells and cells of the immune system. Okay, now here are the cells of the immune system. If we start thinking about that, these are the hematopoietic stem cells. Now hematopoietic stem cell means the poiesis means the development of heme means presenting the iron, so it is definitely talking about the blood cell. So it's hematopoietic stem cells. As they are stem cells, they can give rise to many different types of cells, so they are totipotent in nature. These stem cells can renew itself because it's a self-renewal ability as well as proliferation of other cells. Now the proliferation of two different types of cells can be obtained from a hematopoietic stem cells. They are myeloid progenitor and lymphoid progenitor. Now from the myeloid progenitor, they can give rise to many different types of cells according to the presence of different pigments into them. We can divide them granulocytes. Okay, so what we can find here in this case, from the myeloid progenitor we can have granulocyte, monocyte progenitor, eosinophil, basophil progenitor, megakaryocyte, and erythroid progenitor. On the other hand, from the lymphoid progenitor we can have two different types of cells. One is a uh, lymphocyte progenitor and another is dendritic cells. Now dendritic cells are also being produced from the myeloid progenitor. So remember this thing, these dendritic cells can be produced from both kinds of progenitor, myeloid progenitor as well as the lymphoid progenitor. On the other hand, this lymphoid progenitor can give rise to T cells and B cells. Okay, T cells and B cells. On the other hand, this myeloid progenitor can give rise to the granulocytes like monocyte, not lymphocyte, because lymphocytes are produced by this lymphoid. So monocyte and basophils, eosinophil, megakaryocyte, which can give rise to platelets, and also erythroid, which can give rise to the erythrocyte cells. Okay, erythrocyte means the RBCs. Another common name of erythrocyte is RBC or red blood cells. Okay, so it's all from the myeloid progenitor. We are not going to talk about red blood cells much in our discussion. We'll be talking about the leukocytes or white blood cells in our discussion. And these granulocytes, for example, eosinophil, neutrophil, basophil, these three type of cells as well as T cell, B cell, all of them are among the white blood cells or leukocytes. On the other hand, these platelets are different. They are helping us for the blood coagulation system and so on. Now let us first talk about monocytes. Now the monocytes are larger molecules. The monocytes are larger, not molecules, larger cells, sorry. Now here you can find, if you stain a blood smear with different stains, now you can it can stain like this. Now because it is having granules of different chemical substances, it can be colored like this pink inside the cell. Okay, so here it is. So it's a monocyte, and this monocyte is looking like that. Now this monocyte is having particular bilobe nucleus, not fully cleaved, but still it is looking like this, and then lysosomes are there and all the other cells are there. Okay, now these monocytes can be converted into a large kind of cells when these monocytes enter from the blood cells to the tissue. Now they are circulating in the blood cells all the time, but when they invade the blood vessel and come into the tissue, they will be converted into some other giant cells. Now the conversion results in two different morphological changes of these monocytes. One is that they increase the content of their cytoplasm, almost 5 times increase in their cytoplasmic content. This is the first case, and the second case is that they also become bigger and bigger, and they start migrating, and they start phagocyting different bacteria or the pathogens that enter into our tissues. Okay, so these two things are making themselves really dangerous. They start producing enzymes which can degrade the uptaken pathogen on all this. Okay, so they'll become macrophage from the monocyte. So this is the monocyte, and this monocyte will become macrophage once it is entered into infected tissue. Now, infected tissue can give rise to different signals. Those signals can attract these monocytes to come there and become macrophages. Now, here you can find the activity of the monocyte. So this macrophage will come and engulf the bacteria. It will digest it with the help of the fusion of this. You can see the fusion of the lysosomal enzymes as you can find here. Okay. Now this is an example of another type of dendritic, another type of antigen presenting cells. We'll be talking about this later, the why they are called antigen presenting cells. Now this is a dendritic cell. We have seen the rise of dendritic cells. It is from both type of progenitor, myeloid as well as lymphoid. Now these cells, they look like dendrites or dendrons. That's why they call dendritic cells, because they are having lot of these arms coming in and out. Okay. So these dendritic cells can also phagocyte pathogens, and they can also represent the part of the pathogen onto their cell surface. That's why they call the antigen presenting cells. Okay. Okay, change the color, please. Okay, fine. Okay. Now here you can find the granulocytes, three types of granulocytes. We have seen eosinophil, neutrophil, and basophil. Now eosinophil, this is the eosinophil, neutrophil, basophil. Now one thing about the basophil is that basophils can be converted into another type of cells when they encounter particular chemical signal. They are called the mast cells. When the basophils start granulating, they start producing small chemical substances and can release them. We call them the mast cells. Now these mast cells they are responsible for the allergic reactions in our body. We'll be talking about these mast cells and the role of this mast cell in the allergy production later. Okay, but these cells are very very important during this allergy or hypersensitivity reactions. Now let us talk about the lymphocytes. Now among the lymphocytes, remember we have talked about there are three types of basic lymphocytes. One is the natural killer lymphocytes. Okay, or they are a typical type of T cell. Okay, uh, so natural killer lymphocytes. So natural killer cells, there are two types of. One is natural killer cells, which are come to existing from the lymphoid progenitor. Another one is T cell, a cytotoxic T cell, which is also natural killer type, but it's not exactly the natural killer. So T cell can be divided into uh, two different parts. One is the T killer cell or cytotoxic T cell, or TC. Another one is a T helper cell, or helper T cell, or TH. On the other hand, the B cells are of one simple type, but according to their presence and the activity mode, we can divide both T cell and B cell into two types. One is a normal or natural T cell or B cell. Another one is the memory T cell and B cell. We'll be talking it later. Now the function of natural killer cell is that they are small, not small, but they are cells just wandering around throughout our blood systems and tissues. Whenever they encounter pathogen, whenever they encounter a pathogen infected cell, what they do, they just come in, they just check for the pathogen infection. If they found that there is a pathogen infection inside the cell, they start releasing some enzymes, some chemical substances that can go and kill the cell and destroy the cell. Okay, destroy the infected cell. On the other hand, they can also destroy, most of the time they destroy the infected cell, but not directly destroy the causative agent. And this T cell and B cell, among the T cell, cytotoxic T cell, this T cell are working like the same manner. It will check whether a cell is healthy or not. If the cell is not healthy, it will kill the unhealthy cell. Now why they at all require for doing this? Because remember, there are two different kinds of infection can happen inside our body. But there are two mode of by which bacteria or viruses can infect our cells. Actually, there is one mode for viruses, but there are two or three mode for bacteria to inside our cell. Okay, now say this is our cell system. Now inside our so these are the cells of our body, for example. Okay, and this in between regions, so change the color first. Okay, now this internal regions are interstitial fluid. Okay, interstitial fluid. And these are the cells. Now one bacteria, if I change the color for bacteria here, sorry, let me take it. Yeah, green. Okay, now if a bacteria resides, it can reside inside this cell, or it can reside in the interstitial fluid. So this concept is very very important, so never forget this concept. Try to internalize this concept very strongly, because it will help you to understand the whole concept of immunity. That there are two possibilities for this bacteria to reside. One is that this bacteria can invade to our cell and can reside into our host cell, or other possibility is that this bacteria can stay at this extracellular fluid, okay, interstitial fluid. Now if the bacteria resides into the cell, for taking care of this kind of cell, they will make the cell damaging. Now this natural killer cell can look for these cells, this type of cells, and also cytotoxic T cell can look for this kind of cells, that those cells are damaging, those cells are unhealthy. Now if they find this kind of cells, they will kill these types of cells. Okay, but on the other hand, if the bacteria resides outside, we need other types of cell to fight this infection. And those cells are macrophages. Those cells are neutrophils. Now, for calling upon those neutrophils and macrophages, we need the help of some other cells, and that is this T helper cells. Okay, and also for killing this kind of bacteria which are just freely living on the interstitial fluid or extracellular fluid, we need the activity of B cells, because B cells are the kind of cells they can deliver immunoglobulins or antibody. So T cells most of the time go against the cell with the pathogen, so they are going the cytotoxic effect, but B cells are giving the humoral effect or the liquid effect or the antibody effect on the immune system. Okay, and if we look at here, this is the small resting lymphocyte. This lymphocyte can be produced, this lymphoblast. It could be of two different cells. It can give rise to two cells, effector T cell and then plasma cells. Now B cells are also called plasma cells because these cells, actually B cells can be converted into plasma cells during the maturation. Now each of these cells like T cell and B cells, there is a start point as a progenitor cell. Then as the cells start to mature, it will become much more larger and much more productive. Now those larger cells, productive cells, will start producing different components for proper functioning of the immune system. For example, after the proper large enough of the B cell and maturation of the B cell matured into the plasma cells. Now the plasma cells are much more bigger in size and shape than the B cell at the starting point. Now these plasma cells will produce lot of protein producing machineries or protein synthesis machineries to finally generate what is called immunoglobulins or antibodies. Now this antibody will be produced by these B cells, and B cells can throw these antibodies outside the cell, outside the tissue layers or the interstitial fluids or extracellular fluids, and it can go and grab these bacterial cells and can easily kill these cells. Okay, now if we look at the normal cell adult blood counts of the different cells, we can find the red blood cells are the most abundant one, then the platelets and the leukocytes. Okay, so the actual goal for me for understanding that here, this comparison, so in any kind of blood system, platelets are even in higher amount than the leukocytes. Okay, so leukocytes mean the white blood cells, WBCs, remember. So WBCs presence is much more less than the platelets. That's a very important part you must know. And we are having different cells, and these are the functions of the cells. So macrophages, they phagocytic cells, they can engulf bacteria and other organisms that can cause diseases, and they can also cleave their part and can present the part of this pathogen onto the surface of the bacterial cell. That's why they are called the antigen presenting cells. Dendritic cells can also engulf the bacterial cells and viruses, and then they can present it by chopping the part of the cell out and can present this antigen. Antigen means the foreign particles. So the part of the foreign invader that invades our cell, when they chop this part out and hold on to it, it is called the antigen. And a good material to fight against this kind of antigens are called antibodies, which are produced by the plasma cells, which are just maturated phase of the B cells. So dendritic cells, as they are presenting these antigens, they are also called antigen presenting cells, but these dendritic cells present antigens in the lymph node. So they are called the follicular dendritic cells and lymphoid dendritic cells. Neutrophils are very very important. Neutrophils are phagocytic cells, but they are not antigen presenting cells. So they are not antigen presenting, but they are phagocytic, and they are very very important cells because they present in the blood. So as soon as they find the pathogen, they start engulfing them. But remember, macrophages are hard to achieve. They are scary guy. They are dangerous, but they are hard to achieve because macrophage is not the start point. The start point is monocyte. So when the in the blood it is called monocyte, but when after the activation of this monocyte, that there, if there is an infection inside our tissue, there are chemical substances that can go and give the signal to monocyte to become macrophage and come in. So the activation of macrophage takes some time, but neutrophil can act pretty faster. Okay, now eosinophil excuse me, eosinophil is a very important part which can help killing the antibody coated parasites. There are parasites like worms can infect our body. Now eosinophils can go against these worms via a process called uh, antibody mediated cellular cytotoxicity or something like that. Okay, ADCC. Okay, yeah, antibody dependent cellular cytotoxicity. So this is by the eosinophils. We'll be seeing the process of this killing later. And then the basophils are something which will be converted into mast cells. Now actually those are pretty similar. Now they will be converted into this part. Okay, so these basophils and mast cells, they release the granular components which contains enzymes like histamine and other active agents. Now this histamine and other active agent can cause different type of physiological symptoms like vasodilatation as nasal region constriction. It can sometimes release substances can sometimes lead up to the death of the organism. So it is very, we need to be very careful about the degranulation of the mast cells because it is having a lot of granules inside it which contains the enzymes and chemicals like histamine. Now, if we look at the organs of immune system, we have talked about the cells of the immune system. Then we'll be talking about the organs. So up till this part, we have talked about the cells, and I hope this will help you to learn about the cells of the immune system. Thank you.